would like to introduce with this slide just stating that I think it's quite easy to diagnose CLL nowadays, especially with the help of immunophenotyping. But it's difficult to judge on the prognosis. You've seen that the prognosis can range from months to decades even. And so this is actually the major challenge we're seeing today. And as always in life, things sometimes look very equal, but they aren't. And so this is the statement: not all patients with CLL are equal. Just pulled out this from a dog food campaign, and to be politically correct, it's also man on this. So this is the challenge we have to face. There are a lot of prognostic markers. You see the plethora here, so a lot of things: stage, age, gender. I don't have to read through all this. We will discuss some of these items later on. Actually, I have borrowed or stolen this slide from Emily. It's the Spanish or the Catalanian CLL population, showing some the same difference we expect between BNA stage A patients with a life expectancy of about 15.5 years down to only 3 years for BNA C patients. And the thing is that the majority of patients, they are in BNA A. So it's about 70 to 80% of all of our patients are in BNA A. So if we assume that you don't have to treat BNA A patients, and this dogma is correct, then we could all retire because only 20% are left. But this is, I think, nevertheless, a challenge, whether we have to do an early intervention for this majority of patients or not. And one of our colleagues, Dr. Dijero from Paris, he's now in South America, retiring actually, and he was getting old over this question because he had to wait for a long time until he could solve this question. So whether chlorambucil adds something to the life expectancies of BNA A patients or not. And you see the clear-cut result does not add anything. You can argue now that, in the 90s, there was, not these kind of prognostic markers we wished to have. So this gets us to the topic of this talk. So coming from Germany and being member of this study group, the German CLL study group, I can tell you a little bit from the early days in 1996. Michael Halleck and myself, we were thinking about how could we deal with these early stage patients. This was in the Munich time. And in the mid-90s, we had very simple markers like bone marrow infiltration, lymphocyte doubling time. It's very cheap. A little bit more expensive was thymidine kinase because you have to use an immunoassay, and the beta-2 microglobulin. So, we made a risk stratification out of these 4 parameters. It's here on this left side, and stratified between low and high risk. And only the high risk population was randomized between fludarabine treatment, which is nothing, watch and wait. Then 5 years later, about 2000, we included also other things popped up. So we included genomic markers, we included CD38 and sub-70. And this was a trial that included about 900 patients. So it's also a lot for us and took some years. These are some unpublished preliminary results out of this. You see that we could separate the, maybe let me use this laser pointer, We could separate the low risk from the high risk group by the survival plot. And we see that the monotherapy with Frutara adds something to the prognosis of the high risk patients. So you can see this p-value, but it's not earth-shaking. So adding Frutara, the PFS went from 24 to 32 months. So I think what was more important for us that we could separate by using these prognostic parameters like lymphocyte doubling time, thymidine kinase, etc., that we could separate these two risk groups. Further on, these are now here, the signal data, thymidine kinase, lymphocyte doubling time was useful, the cytogenetics that were included, but these were not stratification parameters, and the VH mutational status. But what was not significant was the bone marrow infiltration pattern and the beta-2 microglobulin for these early stage patients. So the conclusion was we have to work on a new concept. And this new concept was mainly based on the introduction of fish cytogenetics in the field, and especially Hartmut Döhner and Stefan Stögenbauer from the Ulm group. They introduced this technique in our study group. And so these are the hallmark data from the Ulm group regarding cytogenetics. You see it's an independent prognostic parameter for all stage A to C patients, but especially also for the BNA patients. And actually more than 50% of patients in this New England Journal study were BNA patients. You see the high-risk population, the 17 P minus patients, followed by the 11 P minus deleted patients. You might know the dogma that there are no translocations in CLL. We looked back in the literature. There was some old reports by Ulison from Sweden, and we wanted to know whether we are missing something by just using interface FISH. So we resuscitated this method, and the major problem was to induce mitosis in CLL cells in order to get some metaphase cytogenetics. And actually, my colleague, Dr. Christine Meyer, she used to work in my lab. She induced mitosis in CLL cells by just using a CD40 ligand feeder and also CpG plus IL-2. And she received metaphases in more than 90%, actually 98% of samples. And by this, she was able to do these metaphase cytogenetics, and in more than 30% of samples, she received translocations in CLL cells. This is just one example. You see not only 2 partners, but sometimes you can detect 3 partners in translocation. And we had to prove that this is not an artifact, so that we really don't induce, just by our stimulation, some nonsense in these data. And so we looked in unstimulated CLL cells with very specific markers, so in this case FISH markers, and we see the same results. Also here we can, for example, here deletion 5 or derivation 1718. This is not only fooling around with molecular tools. This is, we think, although it's a retrospective analysis, I think could have some meaning in the field. So just looking for the probability of treatment-free survival, we can separate patients with translocation. They're doing quite worse compared to patients without translocation. And we can separate, for example, the 13q population just by using this method. Using a multivariate analysis, including also the BNA stage, the occurrence of translocation was even more important than the old BNA stage. But this has to be confirmed in prospective randomized trials. Oh, I just switched off, sorry. There, 2 years ago, there was a small publication in leukemia just showing using translocations you can predict treatment failure. So we hope that there are also other groups looking whether this has further importance or not. We checked using microarrays whether we can detect different gene profiles using samples with and without translocation. And indeed, we can separate two groups. And I can tell you that, for example, MYC target genes, they are especially prominent in the translocation plus population. Emily has talked about the Ig mutation status. I think this is, besides cytogenetics, the very important prognostic parameter in CLL. And you've seen this model. You now know what the mutated CLL means, and that we separate it from the unmutated cases. And so I don't go into detail again on this. You have also seen these two trials. These are independent trials being published almost at the same time. I think it's very important to realize the prognostic power of these parameters. The median survival is 8 to 9 years in the unmutated cases, but it's more than 24 years in the mutated case. So it's an enormous difference just by using this, this marker. You see, you don't have to read it because it's almost not readable. There are a lot of, especially in the back row, there are many publications about the age mutation status and its importance regarding overall survival. But there are some drawbacks. Dr. Montserrat pointed out already there are few exceptions of the rule. The VH321 gene usage. These cases are usually mutated, but they behave like unmutated cases, so have a very bad prognosis. They usually show p53 dysfunction. And there's a lot of discussion about the cutoff level. Should it be 90, 98, 97, or 95%? What about the choice of database you're using in order to define the germline sequence? There are 3 different databases, and if you look in the trials, they use all different databases. And we also know that 5 to 17% of CLL clones express more than one functional Ig heavy chain. So it's not as easy as initially thought. And we already heard it's laborious, expensive, time-consuming, and it requires special equipment. So it's not routinely done in every lab. So people were thinking about surrogate markers. And CD38 seemed to be one very attractive surrogate for IgVH mutational status. See here the Jelinek publication showing nicely a separation of curves. If you have high CD38, then high CD38 is the bottom line, then the probability of survival and progression-free survival is inferior. But within the same trial, you see they use different cutoffs, 20 and 30%. So the question is it a good surrogate for IgV mutation status? And I think it's has been shown that the association between the mutation status and CD38, it's not absolute. And we also know nowadays that expression can vary over time, up to 1/4 of all cases. So it's not stable. And we have seen that there are different cutoffs used. And there's also something called a bimodal CD38 expression profile. That means you have within this clone variations. It makes it even more complicated. And this happens in up to 28% of cases. So the conclusion would be, from my side, the CD38 cannot be considered as a very good surrogate marker for IgVH mutational status, but it can be very useful because it's an additional prognostic marker. It provides information, although you see from the technical point of view, further standardization is required. You've seen this picture before, so you should have a déjà vu. These are the data from Andreas Rosenwald. Actually, he's also German. He spent some time at the NIH, but he's now back in Würzburg in Germany. And he showed that especially the VH unmutated cases, they have a high sub-70 expression. And this was also confirmed by the group of Wendtner 3 years later. And you see here the data from a famous group in Barcelona, actually headed by Emily Montserrat. He was the first who used this very important prognostic marker on a protein level. So he used the flow cytometry method using a 20% cutoff. And you see that the sub-7 positivity is seen in unmutated cases. And there are only 3 out of 56 patients with discordant results. So this was one of these important hallmark papers regarding sub-70. You see that sub-70 expression translates also in prediction of progression and also likelihood of survival. But 1 year later, in the same journal, in the New England Journal, there was a publication by the group of Thomas Gibbs, recently published that there are about 1/4 of discordant cases if you look for the link between sub-70 and the VH mutational status. So I think my conclusion on this is sub-70, it's a strong marker, but there's no absolute association with the IgVH mutation status. It ranges from 65 to 83%. It also may change over time, so it's not stable. For example, if you use corticosteroids, you can see a decrease in sub-70 expression. So there's a lack of standardized methods so far. There are a lot of efforts worldwide, also from the ERIC, from the European Research Initiative. And so far, I think the best method to be used is the proteins or the flow cytometry methods initiated by the Spanish group. There's discussion about the sampling, should be fresh samples, about the antibody, the fluorochrome, etc., etc. You see this. So I think sub-70 has a very strong prognostic information, especially complementary to CD38, but it does not serve as a very robust surrogate for the IgVH mutation status due to some unresolved issues mentioned before. So where are we standing here? So going from the mutation status, I think there's again this very large discrepancy in survival expectancy. And mutation status is linked to cytogenetics, so unmutated to poor risk fish. It's also linked to CD38, and it's also finally linked to sub-70. So we thought might be useful to integrate all this information in one trial. So we initiated the CL7 trial, not by ourselves, but by together with our French colleagues. And we wanted to know for the large population of BNA patients using these old markers, lymphocyte doubling time and thymidine kinase, plus the new ones, the IgVH and cytogenetics. And again, we assess some other markers like sub-70 and CD38, but these are not certification markers. Then we could based also on our old trial, the CL1 trial, we could define risk. And low risk was defined if you have less than 2 out of these 4 markers. And high risk is defined if you have at least 2 out of these 4 markers. And these high-risk patients were randomized between 6 cycles of FCR or watch and wait. And I can tell you now some early results about the recruitment. You see, besides Germany, it's France, Austria, and Switzerland. And last year we had 629 patients in. And actually, I can tell you, yesterday we had 703 patients in, so we are speeding up. And about 1/4 of patients is a high-risk population. And you see 63 out of these 130 patients received FCR, and 67 were just watch and wait. So this is the interesting group we have to follow for the next years. But there are also activities. John Bird, probably he won't talk about this by himself because he has another topic. But, you know, by the CLGB, there is a stratification just based on the mutation status. And to my knowledge, this trial was initiated in September 2007. So this is the US approach. There's also a British approach, just using these 3 markers, including CD38. And the British way, they separate the 17p as a separate group and did a separate randomization for this. So all these 3 trials are running, and we're waiting results. Now I'm coming to something very German. It's the inclusion of comorbidity in our trials. And so the question is, should comorbidity use instead of a calendrical or a chronologic age as prognostic parameter? But this goes back to a lady living in South Florida. It's Dr. Exterman. She introduced the so-called SEER score. It's a cumulative illness rating scaling. And she demonstrated that the highest SEERS rating is linked to inferior outcome in cancer patients. We don't know the single impact of these single factors, but nevertheless, we thought it's worthwhile using this SEERS scoring, also in our trials. And you see here a 14-bullet questionnaire. This has every German physician has to fill this out when he wants to recruit patients in our trials. So what we calculated and stopped, it's only a 2-minute work. So he has to put some points here ranging from 0 to 4. And if you're ending up with a score above 6, your patient is considered to be frail. And this is also been shown by Dr. Exterman before that 6 seems to be a reasonable cutoff. And so we have these new words. If a patient is frail, we say it's a slow-go patient, and it ends up in a no-go patient. But if it's a fit patient, we call them a go-go. We do have now from our own trials some data based on more than 500 patients showing that comorbidity, so an inferior overall survival, is especially linked to comorbidity. And you can use different systems like the Charlson Comorbidity Index also, the same comes out. And if you put in a multivariate analysis, also age is non-significant. It's in this case, the Charlson comorbidity scaling, which reaches the significance level. So the impact of comorbidities on survival is independent of the patient's age. The comorbidity is superior to the calendrical age in predicting survival. So these are the conclusions from this. Dr. Montserrat briefly mentioned the MID negativity, so also we'll only briefly go into detail with this. And it's besides the Barcelona publication being published in Clinical Cancer Research last year, there's this important paper by Merton from 2005 showing if you use Campus up to MID negativity, patients really survive longer. So there seems to be some impact of MID negativity on overall survival. I will show tomorrow in detail by discussing the CLA trial, but this is some, slide preview on this. If you use also the MID negativity in a modern trial, you can show if you reach MID negativity, you just have a longer progression-free survival. And this is independent of the treatment you get, either FC or FCR. But the whole story is the probability that you get MID negativity is higher in the FCR arm compared to the FCR. But we will talk about this tomorrow, and after all, it's only PFS and not overall survival so far, so we have to wait longer on these results. There are also some more experimental prognostic markers, so I would call this work in progress. Some of them is house-made. the lipoprotein lipase. This seems to be, you can see from this list of publication, also something interesting. It can be easily measured by PCR, and it's relatively stable. So in contrast to some of the other markers I mentioned before, so CD38, for example. And it seems to have also some functional role based on fatty acid metabolism and cell plasticity. My own research group, we've done some experiments stimulating CLSs by the BCR, and we could show that you can increase LPL expression by this kind of stimulation. Another, just scratching this parameter, interesting marker for the future maybe is Tozo. It's named after a Japanese sake. They usually drink it for New Year's. They believe they live forever. Maybe it's a Japanese way, but this name was also given to this molecule because it's an inhibitor of apoptosis, and we could see that in CLL it's exclusively expressed compared to other normal B-cell compartments. It's also exclusively expressed compared to other B-cell malignancies. And these are just some lymph nodes. In the bottom you see the red ones are the Tozo positive CLL lymph nodes. And for example, follicular lymphoma, they are negative. And there's also some link to other prognostic parameters like mutation status and staging system using Tozo, but we have to wait longer for this and check this in a very meaningful, randomized way. Briefly, also microRNA, another marker. I think it's very early for this marker to be introduced in the clinical routine. There's some publication in the New England Journal about the link between microRNA signature based on these 13 microRNAs and sub-70 and mutation status. They could show some survival curve differences using some of these markers, but I think it's far away from being applied in the daily routine. So it comes to the question, how can we make use of all these prognostic markers in the clinical setting? I think the definition of a poor risk CLL patient in the year 2009, it's a male, he's older, he bears some high-risk cytogenetics, shows an unmutated IGVH mutation status, it's sub-70 positive and CD38. But can we use molecular markers to decide therapy? I think the general answer is no, as Dr. Mossera already told. Maybe there's one exception that if we look at the 17p- defects on the very high-risk profile. Looking back in our trials, the CL1 for BNA, the CL4, the F versus FC, first line in the younger ones, and the CL2H, this is a refractory population, you see that the rate of P53, the percentage of P53 positive patients increased from 0.5% up to 35.6%. So these are trials. You can conclude for the daily life that at least in advanced disease you will get an increased percentage of P53 defects. Now, going for overall survival in this population, we know we can't rescue them by F or FC. These are the survival plots. You see the bad curve for 17p- in the CL4 trial, and there's no difference whether you check for mutations, P53 mutation, or just for the deletion. So it's the same. This data was shown by the Stögenbauer group last ASH. And also, you will see this tomorrow again, overall survival for the CL8 trial. We can equalize prognosis for almost every cytogenetic subgroup, but we can't do this for the 17p-. So this stands out. We know that using Campeth, we can at least achieve a similar overall survival in the relapse setting using this antibody for all cytogenetic subgroups. But I think nowadays, the final answer for these kind of patients is a transplant. 17p- we consider as a very high risk. And so this is done within the CLX2 trial, and we will hear tomorrow afternoon from Dr. Montserrat more about this. So I will close up. The IWCLL guidelines, they show some recommendations, but they say it's desirable to use cytogenetics as a prognostic factor. I can tell you in Germany, we just do it, but this was an international consensus. So, after all, I think the most important thing is that we should base treatment decisions on the clinical staging systems, as told before by Dr. Montserrat. And so I would like to conclude. I think results of large prospective clinical trials evaluating possible long-term survival benefit of different regimens, they are still lacking. So this is the first one. Second, I think we have seen several prognostic markers that require further validation and also standardization. And for the time being, I would say international guidelines for treatment initiation will remain based on clinical criteria indicating disease progression. So the BNA or RI system, for example. And the future goal remains to reliably predict prognosis, diagnosis, thus enabling risk-adaptive therapy. That's something we all wish for individual CLL patients. The German answer to the prognostic factors is we base our trials on risk, stage, and fitness. You see here the go-go again for the CLL10 trial and the slow-go for the CLL11 trial. I will tell you more about this tomorrow. And I would like to thank my colleagues at the headquarters in Cologne of the German CLL Study Group, but also of our co-workers on the cytogenetic part, Stefan Stilgenbauer and Hartmut Döhner, and for the MID part, the Kiel group headed by Michael Kneber. I would like to thank my colleagues in my lab in Cologne. And finally, because Wendy asked me I should show her some pictures of my kids, I didn't have them with me. I would like to thank my two kids that they let Daddy go overseas for these days. Although they like the snow very much, but I will go skiing for them tomorrow. Thanks.